Thanks everyone for joining us this evening. My name is Eileen Anderson. I'd like to welcome you and thank you for joining us, whether you're participating live this evening or if you may be viewing this at a later date, because we will be posting the video online for you to share or to come back and review, or maybe you'll just be logging in later to be able to check it. Just a few words before we start. Our job in the Sue and Bill Gross Stem Cells Research Center is all about the science of regenerative medicine, from very basic discoveries all the way on up to clinical translation and clinical trials through our Alpha Stem Cell Clinic. But even more broadly, just checking if I have a video problem there. Even more broadly at UCI, we're really about education, educating undergraduates, graduate students, medical students, and so on. But the COVID-19 pandemic has really highlighted a different role for academic institutions like UCI, where we can serve as an engine for outreach, for guidance for the broader community, to answer questions, help people understand really what's going on, and to be a nidus for innovation and for solving problems, addressing new technologies and platforms to help us face the challenges that we're all facing right now as a society. The Stem Cell Research Center was particularly glad to be able to step up and assist with producing some of the medium that was required in order to jumpstart testing in Orange County earlier this year. And that's partly because a number of our faculty have been so closely involved in COVID-19 and clinical trials. And so I'm going to come back and tell you about that in just a moment. I just want to, interesting. Pause 1 second because I am unable to advance my slides, but that's okay. We really don't need them. I am grateful tonight to have been able to organize a follow-up to the COVID-19 information panel that we hosted in earlier this year on May 14th. We have 2 colleagues from within the center and 2 guests from the UCI School of Medicine that are here to address 4 topics of interest, I hope broad interest for the public. First, an introduction to the virus that underlies this pandemic. Second, an update on COVID-19 testing and what our standing is for that. Third, a discussion of back to school, masks, public health issues that all of us are very concerned about right now. And last, some perspective on clinical trials for COVID-19 and how those clinical trials in reality relate to stem cell technologies. A few organizational bits before we go ahead. I will introduce each speaker and they will do short individual presentations on those 4 topics. Please do send any questions that may come up for you as we go. You can use the Q&A box, which should be to the bottom right of your screen in order to do that. We have also collated questions that have come in during the course of the registration and allocated those as much as we can, box them together so that we can be efficient in terms of sending them out to the speakers. I will moderate a mix of those collated questions and the live questions that come in via the chat and ask the speakers to respond. Please do understand if your specific question does not get asked, it may get condensed with a different question that's similar. And also, if we should for some reason not get to the specific question you asked, we will be putting together a set of answers for those over the next day or 2 and posting them online. So if you feel that your question didn't get answered, you'll be able to check back online and see if we've been able to get to it. So we should be able to do that. Last of all, I'd like to say just a couple of words of thanks in terms of putting together this meeting. We normally host an online forum, a community lecture series once a month, which we've done for almost the last 3 years at the Stem Cell Research Center. I would normally be telling you about all of our exciting research, the status of where those things are, how we see ourselves going forward. But again, our role here tonight is really about providing information that's much more granular and relevant to what's going on in people's lives right now with COVID-19. I do encourage you to visit our website where we have those past videos posted. And I also want to highlight 2 dates that are coming up. I'll show those slides at the very end as a reminder, but you can find them on our website as well. September 14th and 15th will be a jointly hosted meeting between UCI and the UCI Stem Cell Research Center and UCSD. That is the grantee meeting for the California Institute of Regenerative Medicine. It is open to the public. It is lay friendly in terms of the talks and the panel discussions that are going to be held. And it's an opportunity to understand where stem cell research is in California and get an update on what happened really with Prop 71 and the ballot initiative and how stem cell research has been invested in by the state. The second thing that I want to highlight for you is October 6th, we'll be having a Prop 14 panel discussion hosted by the center. This is going to be an opportunity to ask questions about the upcoming ballot initiative with neutral parties, with people that are pro and people that are con from across the state. And so I encourage you to have a look for both of those things. With that, I'd like to introduce the first of our speakers. This is Ming Tan. He's going to be giving us an introduction to SARS-CoV-2 and COVID-19. Ming received his bachelor's degree from the University of Florida and his MD from Johns Hopkins University. He is a professor of microbiology and molecular genetics and an expert in infectious disease. And Ming, I'd like to invite you to share your screen and go ahead and start your presentation.
Thank you, Dr. Anderson. So this is a virus that's now become all too familiar to us. The name is SARS-CoV-2. As you know, the infection that it causes is called COVID-19, and colloquially we often just refer to it as the coronavirus. But there are many other coronaviruses. This is a family of viruses, very familiar to us, in fact, because it's a common cause of the common cold. And so about 10 to 30% of the cases of common cold are caused by coronaviruses. So we've all been exposed to coronaviruses in our lifetime. Now, this particular coronavirus, SARS-CoV-2, is different. We think it might have come from bats. It is called a coronavirus because if you look at this highly magnified image of the virus, you can see that there are these spikes sticking out from the virus, and those form a crown-like appearance. And crown, the word for crown is corona, and so that's why it has the name coronavirus. Now, importantly, these spike proteins are used for attachment to human cells. And another important part of the virus is that within the virus is the genetic material, which is RNA. A lot of organisms, the genetic material is DNA. In this case, it's RNA. Now, to put the COVID-19 pandemic in context, this is really the second most important pandemic in the modern era, and it has the potential to become the most important one. So the frame of reference, of course, is the 1918 influenza pandemic. It's estimated that 50 million people died, although you'll see numbers up to 100 million, and also estimated that 35% of the world's population was infected. So for context, for COVID-19, and we're not quite done there, we have 850,000 deaths in the world. We're approaching 1 million deaths. 25 million known cases, which is 0.3% of the population. The problem is those are the known cases. We don't know how many people have actually been infected. Some early studies have indicated that the total number of cases is often 10, 20 times more than the known cases. So this number may be large, and certainly larger than 0.3%. Is it quite 35% yet? Probably not. But again, we may be in the early days of this pandemic still. In the US, the 1918 influenza pandemic took 2/3 of a million deaths. We're already up to 180,000 plus deaths. We'll soon be at 200,000. So it's not quite there, but certainly at the level where this has been a huge impact on many people and affecting all our lives. Now, in the US, about a quarter of the population were infected in 1918. We don't know the exact number. The known cases in the US have now surpassed 6 million, which would be 2% of the population. Again, the actual number is definitely much larger than that. Now, if we look at the 1918 pandemic, it famously was known to occur in 3 waves. So there was the initial wave in the summer of 1918, and then it came back with vengeance in the fall of 1918. And on the y-axis, we have the number of deaths per 1,000 people. And then it also came back in the spring of the following year, 1919. With COVID-19, we really haven't seen distinct waves, but there was concern initially, was it going to be seasonal spread that occurred at certain times in the year? So far, we've just had a continuing pandemic that hasn't gone away. But again, these are different viruses. We don't know if the behavior will be similar or different. So this is a graph from an online site called Our World in Data. I find it very useful. You can look at many different things. In this case, I'm looking at new cases, new daily cases for different countries. And you can see with the US, we did flatten the curve, as the expression is, but the curve didn't come down. There are countries like India where the curve is still rising, in fact, now surpassing the United States. So we're not the country with the most daily cases anymore. There are countries like Spain that initially had a large outbreak, thought it was contained, and now cases are going back up. South Africa, and like a lot of Africa, has had a late start, but they seem to be coming down. And then China, we know it started in China. At some point, they were reporting no cases, but now there's some cases again. So what we're seeing is that different countries have different responses, but there now are indications that even if you have a good response early on, the virus can come back again. This is another way to look at the global impact. So this is looking at deaths, daily deaths, and you can see that we had an initial peak here when this was in April, May, when there were lots of cases in Europe and also in New York City in North America. If we fast forward to where we are today, we still have a lot of cases in the US. We never really had our cases go all the way down like they did in Europe and Asia. But you can see that South America and Central America now make up a lot of the cases. And that often doesn't get the full attention. And India also has a lot of deaths. The total size of the daily deaths is now approaching the peak that we had in April, May. So back to the US, we have now more than 6 million known cases. Again, these are the diagnosed cases. The actual number of cases is for sure be much more, although we don't know the exact number. Number of deaths is approaching 200,000. We did have a flattening of the curve in June, and then we opened up parts of the country, and then we had a resulting increase in cases. It's now coming down a bit. This map here shows current hotspots. So the closer to red, the more daily cases. You can see now the hotspots are in the heartland, in the Midwest, in the South and South Central. Closer to home, we have almost 50,000 cases in Orange County and almost 1,000 deaths. We're averaging about 300 new cases a day. You can see here at the bottom where I've shown hospitalizations and in ICU cases, we had a peak about in July. In July, we were averaging about 1,000 new cases a day. Hospitalizations, ICU rates have started to come down. So perhaps our peak is over, although again, I think what we've learned of this virus is you can never be too vigilant. In the state, we have over 700,000 cases and 13,000 deaths, and the new cases about 5,000 a day. So you can see that California initially did very well, but we never completely had our curve go down, and we've had sustained transmission. One recent change is that we're seeing more cases in children. Now, some of it may be because of summer camps. We have the issue of whether to reopen schools. That will be discussed by Dr. Cooper in just a few minutes. But clearly, we have more cases in children now, more cases, more hospitalizations, and more deaths. There have been some discussion that children don't get a severe infection, but we know that some children have severe infections and there have been some deaths. So a couple of slides now about the clinical presentation of the infection. And so for a typical mild infection, which we think would be about 80% of the symptomatic cases, there is exposure to another person, and then an incubation period, which is the time between when one is exposed and when one has symptoms. And so that typically can be 4 to 5 days, but maybe it's up to 14 days, which is the origin of the 14-day quarantine that you hear about. So symptoms occur, and those typical symptoms would be fever, a dry cough, meaning that you don't cough anything up, and fatigue. Some people can have some shortness of breath, and then other symptoms include loss of appetite, producing sputum, which is the phlegm that you cough up from the lungs, muscle aches, and some patients have altered sense of smell. Generally speaking, for a mild infection, people recover after 2 weeks. And the British Medical Journal is a very good resource if you want to look up aspects of COVID-19 infection. Now, some important details. People are infectious for a few days before they have symptoms. So that's concerning because they don't know they're infected, but they can spread it around. And they remain infectious for perhaps up to 7 days after symptoms. So a total period of infectiousness of about 7 to 10 days. And so again, this is the period here just before you have symptoms where people can be asymptomatic. We actually call it pre-symptomatic. They don't have symptoms yet, but they can spread it to other people. Now, I want to contrast this mild infection with serious infections, which we see in a minority of patients, perhaps 15 to 20%. So they have the same initial infection for the first 7 days. And then after that first week, if someone's going to go on to severe infection, that's when the bad things happen. So they have shortness of breath that gets rapidly worse. They require hospital admission. And within a couple of days, they can actually have difficulty breathing, something called acute respiratory distress syndrome, or ARDS, which can require them to be admitted to the ICU and be on a ventilator. And some of these patients die, and they die typically 2 to 3 weeks after onset of their symptoms. You've heard a lot about asymptomatic infections. So this pyramid shows the symptomatic cases, and then we said most are mild, a small proportion are severe, and the worst cases result in death. What we don't know is the bottom of the pyramid. How many cases are asymptomatic, where the individual doesn't know they're infected? Are we talking about a small proportion, or perhaps a very large proportion? It's been speculated that the asymptomatic cases may amount to 40% of cases, but the answer is we don't know, because it's hard to identify those individuals, at least with PCR testing, which we'll hear about from Dr. Monuki. We're all very interested in whether one can get a second infection, because it's related to the issue of whether there's immunity after you're infected. And so we've now heard some very recent reports of second infections in the same individual. These reports seem to be credible because they isolated material from the virus from both the first and the second infection, and these viruses are genetically different. So there was a case in Hong Kong of a 33-year-old man. He had an initial infection that required hospitalization. And then 4.5 months later, he was screened in an airport. He didn't have any symptoms, and they found he was positive again. This one, where the second infection is mild asymptomatic, is not so worrisome. But there's a case of a man in Nevada who had an initial infection that was mild, but then a month and a half later had a more severe infection that required hospitalization. That's what we worry about, not just being infected once, but will you be infected again and continue to have severe infections? I must emphasize that these are just case reports, isolated cases. We don't know how common reinfection is. We don't know if reinfection is mild or severe. I want to spend a few minutes on transmission because this is important to all of us. So the transmission is human to human. And it's mainly by direct spread from one individual to another by respiratory droplets. So these are droplets which are large particles. They fall to the ground within minutes. We all produce them when we're coughing or sneezing, so symptomatic people obviously can produce them. But even just from talking, from exhaling, famously from singing in a choir, you can produce these respiratory droplets. If you're infected with COVID-19, the virus-contaminated droplets, when you cough them out, if they land on the mouth, nose, or eyes of another person, they can be infected. And the greatest exposure has been 6 feet, although this is a continuum. Now, you can also have spread via your hands. So if I cough or sneeze into my hands and I shake hands with you, I can pass it on to you. If you now don't wash your hands and you touch your mouth or nose or eyes, you can then infect yourself too. There's been a lot of discussion about airborne particles. So these are different from respiratory particles because they're smaller particles. They can stay in the air for hours. So there's some infectious diseases like measles that spread this way, and they're very infectious. Initially, we thought there was no COVID-19 spread this way in the community, but now we have some evidence it does occur. We don't think it's long-range transmission, meaning that we don't think that the virus particles can float around and, let's say, infect everybody in the same room, but we don't know the details of how often this happens. What we do know is that next one. What we do know is that airborne particles are not commonly produced in the course of normal events, but in the hospital where we do things to patients, such as put them in a ventilator, we can produce aerosols, and so there's greater risk for hospital personnel. There's also indirect transmission by objects in the environment. This is why we also practice hand hygiene. And so the idea here would be that somebody has contaminated virus respiratory droplets on their hands, they touch an object, and then you touch that object. This is probably more likely if there's heavy virus contamination that would occur in an infected individual's household or in their hospital room. So to put this all in context then, I think there's all kinds of risks, but I think we need to recognize what are the greater risks and what are the lesser risks, because we're in this for the long haul. So what things do we need to be aware of? So the greatest risk seems to be in poorly ventilated indoor spaces. There have been outbreaks in meatpacking facilities, in bars. In contrast, the risk outdoors seems to be low. The risk depends on the type and duration of exposure. So your greatest risk is face-to-face contact with an infected individual within 6 feet for an extended period of time. The risk of exposure increases the more people you have contact with, and there's relatively low risk for incidental contact. So you pass someone in the street, you handle an object, your risk there is relatively low. How about transmission by asymptomatic individuals? There are two scenarios. One is that the individuals, as we said, haven't had symptoms yet, but they're going to develop symptoms. It's also possible that they're spread by people who never have symptoms. That may contribute to the spread. We don't know how often this occurs. So and for my final slide, what can we do to protect ourselves? Well, wearing face masks makes a big difference, and Dr. Cooper will have more to say about this, but it provides protection to you, the wearer, and also to others. Physical distancing, obviously we're tired of this, but the 6-foot magic rule is because you want to be out of the range of the respiratory droplets. Hand hygiene, limit your exposure, avoid crowds, avoid poorly ventilated indoor spaces. We want to protect the vulnerable. That includes the elderly and individuals who have risk factors for severe disease. And if you have symptoms, the best thing to do is to isolate. And this is going to be more of an issue in the winter season coming up when there are all kinds of colds and influenza. But basically, if you have symptoms, you should stay away from other people. All right, that's my presentation, and I'm going to pass it back to Dr. Anderson. Thank you.
Fantastic. Thank you so much, Ming. With that, we'll move straight into our second presentation. Just a reminder, in case there are a few people, I think there have been, that have joined us since we started, we are going to move from panelist to panelist and hold the questions until the end. I see there are a few that are coming in the Q&A section, again, that's at the bottom right bit of your screen if you'd like to add a question there. We're collating these together, and we'll come back and I'll moderate at the end of all the presentations to be sure that everyone has a chance to ask the things they're interested in. With that, I'd like to introduce our next speaker. This will be Ed Monuki. Ed received his bachelor's degree from MIT, his MD and PhD from University of California, San Diego. He is at UCI, first and foremost, a really valued member of the Stem Cell Research Center here, where he really does fantastic work and is just a wonderful colleague. But he is also the Warren Bostic Professor and Chair of Pathology, Director of the Neuropathology Core for the Alzheimer's Disease Research Center, and the Director of the Clinical Testing Labs for UCI. And so, Ed, with that, if you would please share your expertise on testing for COVID and lessons learned so far. Thanks.
Thank you so much, Eileen, and welcome everybody. I am going to give you an update on COVID-19 testing and talk again about the two different tests that are available. Hopefully I can advance my slides. Great, okay. So here's the outline, and for those of you who may have listened in 3-4 months ago to the first panel, I will try to highlight what has changed in the last 3 or 4 months. And I'll talk about the two tests, more specifically about what a positive test means. I will go over, as I did before, some of the frequently asked questions about the two tests. And then in large part to emphasize sort of the challenges that we and really the nation has faced in bringing up testing, I will highlight what we've been able to bring up for COVID-19 testing at UCI, what our current state is, and what our future plans currently are. So similar to the schematic that Dr. Tan showed about the COVID-19 infection, I will highlight the two tests in context with the infection. So first off, if we imagine getting infected on day 0, and the virus replicates in our body and eventually does get cleared. And typically about 5 days on average after the infection, the symptoms begin to appear. As Dr. Tan mentioned, that can be up to 14 days, however. Then the body and the virus starts to produce the viral products that elicit the immune response in the body and the production of antibodies. First, IgM antibodies, which then tend to wane and disappear from the system. And then slightly later, the IgG antibodies, which persist for some time. So the first test is the PCR test. That's sometimes referred to also as the molecular test or the diagnostic test. And that's really intended to detect the virus in this very early phase around the time that symptoms occur. The second test is the antibody test or serology test. And that is designed to pick up these antibodies that develop later as part of the body's response to the infection. So those are the two tests. So what do those two tests mean, and what does a positive result mean? Well, the PCR test, if you're positive for the PCR test, in this period around the time just before or after symptoms arise, that means you're contagious. Right? And so all of the different public health measures that Dr. Tan mentioned that we should be doing, isolating ourselves, the hand washing, the wearing the masks, the social distancing, those become particularly important in this stage when you're contagious. The antibody test, on the other hand, what it tells you is that you've been infected sometime in the past. And typically, this means that you are, at that stage, not contagious. This is not always the case, but most often, if you are in this later phase of the response and you're positive for the antibody, it means that you're most likely not contagious at that point. Now, one of the things we know actually is, which we didn't know 3 or 4 months ago, is how long this antibody response will last after an infection. We now know that it does last for at least on the order of months, which is good news in terms of thinking about how our bodies might react to the vaccines, for example. Now, one of the other things that we have learned over the last 3 or 4 months is that although this PCR test is meant to detect virus at this point when a patient is contagious, we also now know that this PCR test can be positive for months, 3 to 4 months actually after somebody first becomes infected with COVID-19. And importantly, that positive test does not mean you're contagious. As Dr. Tan mentioned, it's really in those first 8 to 10 days after symptoms that a person is contagious. And in fact, the CDC guidance now is that once you test positive for COVID-19, that you do not actually receive testing for the next 90 days, so about 3 months, just because of this issue that patients can be PCR positive but not contagious. So the recommendations from the CDC with a positive test and symptoms are that you're really not considered contagious 10 days after symptoms if your fever has come down and your symptoms have otherwise improved. So very important thing we've learned in the last 3 or 4 months. So some of the frequently asked questions, in this case, the ones that are mostly yeses for the PCR test. So does it mean you're contagious? Yes. You need to isolate yourself? Yes. Might you need a hospital if your symptoms worsen? Yes. And for contact tracing, which is the idea of trying to limit the spread of outbreaks after a positive individual has been identified. The PCR test is the important test for contact tracing. The antibody test, in contrast, is no for this set of questions. One of the things I wanted to mention that has changed over the course of the last 3 to 4 months, previously when we met, the recommendation was only for this nasopharyngeal swab for the PCR test, and that's the one that is somewhat uncomfortable. Some people have described it as almost feeling like somebody's getting the swab all the way back to your brain. Of course, that's not the case. But it is a very sort of uncomfortable test. Now, we actually, in addition to the NP swab, which is still the gold standard, nasal swabs, where the swab is just from the nostril, the 2 nostrils, or more recently, saliva, and I'll talk a little bit about saliva testing as well. These are also acceptable specimen types for COVID-19 PCR testing. Some frequently asked questions. In this case, the ones that the answers are yes for the antibody test. Does it mean you've had COVID-19 in the past? Yes. Does it mean that you could be a plasma donor? Yes. So this is the test to see if somebody has been infected and could potentially be a donor of convalescent plasma, which is a topic that Dr. Bota will be touching on. The antibody is a test to get insight into the prevalence or how common the infection has been in a community. And then one change, again, from 3 or 4 months ago, is the question about, does this mean there's immunity from COVID if you're positive for the antibody test? 3 or 4 months ago, I had 2 question marks. Now, with recent studies, it seems like the answer is probably yes. Both from different research studies, as well as some of, again, the phase 2 trials that have been completed for some of the leading edge vaccines. And again, that may be a subject that we get back to. Finally, the big question, does being positive for the antibody, how does that impact herd immunity? That is the idea that the entire community has sufficient protection that it prevents or inhibits person-to-person spread. And still the answer to that question is still a question mark. So we don't know. Okay, and then issues with test accuracy, which has been an issue from the start. Still, with the PCR and antibody tests, these same answers apply. Are there false negatives with PCR? Yes. And by false negative, meaning that the test comes back negative, but you actually are infected with COVID-19. And the reasons for that are less about the quality of the tests at this point, but more so just with the timing of the virus, the presence of it only for maybe that first week or so after symptoms. The sampling, we're only sampling typically one site in the nose, and those are the main reasons for the false negative rate. Similarly, the timing of it with the antibody testing, if you test too early, often the antibody test will be negative. And then false positives, typically, again, the PCR test, we don't see too many problems with false positive results. And antibody testing, at least in the beginning, it was definitely a yes and no, because the test quality was quite variable in the beginning, especially for the antibody test. So currently, the test accuracy is better. The tests have gotten better. The FDA has now issued warnings to the manufacturers of some of the poor antibody tests in particular, but also other tests as well. Last time I checked a couple of days ago, the FDA had actually issued over 3,000 warning letters to different companies and makers of these tests. And the FDA now has a pretty robust fraud website as well, up and running. So the situation is better now than it was when we first met. Okay, So what about bringing up COVID testing at UCI? It truly has been unlike any experience I've ever had before, because there have been so many different challenges. And this really reflects the testing world, not just at UCI, but throughout the nation. We all got off to the slow start, so of course we need to prioritize speed of getting testing. Shortages of all different types. Initially, testing reagents and specimen collection kits and the personal protective equipment for the healthcare workers to take the test. Now it's instruments, pipette tips, you name it. There's all sorts of shortages, which means that instead of bringing up just one platform to do the testing, we needed to bring up as many as we could because we didn't know where the shortages would limit our ability to do the testing. The federal diversion, frank interception, and the lack of coordination, particularly at the federal level, requiring us to compete against other labs like we really have never had to before. The bad unapproved test problem, especially in the beginning, which is now better. And the fact that we still don't have an end game or a timeline to the end of the pandemic. And so we constantly need to prepare for the worst. And that really, these challenges really haven't changed over the course of the last 3-4 months as we've strived to bring on more and more testing to test, for example, those asymptomatic individuals, as Dr. Tan referred to. So at UCI, just very briefly, we now have 6 different molecular testing platforms described here, have performed over 100,000 tests on about nearly 50,000 different patients. We have 2 different antibody testing platforms have performed far fewer tests for those on a lower number of patients. And you can see in terms of that idea to diversify, some of the platforms are designed to be much faster, but are much more constrained on reagent availability and throughput, whereas some of the other platforms that allow us to do more volume of testing are slower, but still allow us to generate results in 24 to 48 hours. The last 2 platforms to enhance the volume capacity of our lab were implemented over the last couple of months. This is a snapshot of our lab dashboard, just to highlight a couple of numbers. Overall positivity rate for PCR being about 8 to 9%, and very similar for the antibody testing rate of our patients. You can see kind of a weekly cycle of test volumes, this first line being 1,000, the second line being 2,000, and in the middle of July, we were receiving up to about 2,000 tests per day, and that has started to come down as the test positivity rate has come down and the hospitalization and ICU rates have come down as well, as Dr. Tan mentioned. 3 or 4 months ago, we were able to do about 1,000 tests per day. Currently, we're up to about 2,500 tests per day. In Orange County, as of a few days ago, there had been about over 600,000 tests performed, and at UCI, we had done about 1/6 of the tests in the entire county. The antibody tests, we have done far fewer. So what did that enable us to do at UCI? Well, enable us, in addition to being the first hospital system in Orange County to bring up PCR and antibody testing, it allowed us to reopen UCI Health and the medical enterprise, which is now running at 100% or more capacity on a regular basis, to provide some testing for our healthcare workers and providers, and of course, a lot of community sites, including helping Orange County Public Health as well as Orange County first responders. This is highlighted a little bit more here in terms of some of our hospitals, community hospitals that have asked us to take on some of their testing, including CHOC, the Memorial Care System in St. Joseph's, and doing testing now for over 100 nursing homes or skilled nursing facilities. And then the last slide here, really the next phase is to ramp up testing even further to be able to provide testing for asymptomatic individuals to catch those pre-symptomatic individuals that Dr. Tan highlighted, and first and foremost for our own UCI campus, so they can open to a limited extent in the fall. This will involve one-time testing of current and arriving students, estimated to be about 10,000 students. Thereafter, in the fall quarter, weekly testing of students, so about 2,000 tests per day. And that testing is going to be coordinated at 3 different sites. In the central lab at the medical center, which will involve pooled testing, in student health center, and then in a campus pop-up laboratory where we are focusing on bringing up saliva testing to simplify the process of specimen and deal with some of those shortages of the specimen collection kits that I alluded to. And with that, I thank you for listening and wish you to stay well, and hope that we can all come back and root on our anteaters in person sometime soon. So thank you very much.
Thanks very much, Ed. We do have a lot of questions coming in about testing, so rest assured, everyone, we're putting those together on the side. With that, though, I'd like to invite Dan Cooper to share his screen. Dan, by way of introduction, received his bachelor's from the University of California at Santa Cruz and his MD from UCSF, so he is all in-house in the UC system. He is professor of pediatrics and chief of pediatric pulmonology at UCI, the founding director of the Institute for Clinical and Translational Science, associate vice chancellor for clinical and translational research, and has had the somewhat, maybe not unique, but important experience anyway, of working with a group of pediatric research organized and targeted to eliminate the COVID-19 threat, the PROTECT group, in order to look particularly at issues about bringing kids back to schools. So Dan, welcome tonight. Thanks for joining us, and please go ahead.
Thanks, and I presume everybody can see my slide?
I can certainly see your slides. You might want to share your video though.
Here we go. Well, thanks, Eileen, for inviting, and thanks to Ed and Ming for outstanding presentations. I'm going to talk mostly about the schools and really talk about what the role of our university is in meeting this enormous challenge of reopening the K-12 schools. This is an issue that has occupied much of the news, and I'm sure many people in the audience have children and grandchildren who are facing this question of exactly what to do. And we're part of the Institute for Clinical and Translational Science. We work closely with the Stem Cell Center in bringing basic science discoveries to actually help human beings. I want to focus really to begin with on a tale of 4 small and terrific schools in our region, and we'll learn more about them. This is a photo that was on the Instagram of one of the schools in Santa Ana, California. Many of you know that the schools in Santa Ana and that in Santa Ana in general has been an area where we've seen a remarkable increase in the number of cases and more morbidity and mortality, unfortunately, than in many other communities. And as you know, this is a predominantly the Latinx community and tends to be more lower socioeconomic. This is a picture from super scholar Ruben at the beginning of the pandemic. And he wrote, "Hello to all my Madison School friends. I hope you all are doing well. I miss you all. Hope we see each other soon. Stay safe." And we're also going to talk about our great big university. I like to show this slide because we think of the University of California just in terms of its campuses. But if you look to the right here, we've got agriculture, environmental, natural resources. We have K-12 student services. We have public policy. And so in many, many ways, the University of California is positioned as a public university to address some of the key issues that we've got to face if we're going to reopen our schools. But this is a quote: "The healthy reopening of schools is the most complex, biologically, mysterious, economically profound, and unfortunately politically charged health issue I have ever encountered since graduating from medical school 46 years ago." This was me, actually, my quote. It was in the New York Times Magazine. It's hard to believe that I somehow graduated from UC San Francisco in 1974. This is how people looked back in 1974, in case you don't realize this. But this really is one of the most remarkable problems that I've ever had to face as a pediatrician and as a physician scientist. So why should our university care? So there's mounting evidence that school closures adversely affected health and learning of children and adolescents. The data are increasing to demonstrate this. And it's important in this context to recognize what Dr. Tan pointed out before, which is that children seem to have less symptoms than do adults from this disease. And we don't completely understand that. Fortunately, that's the case. This is not a disease like polio or even influenza, in which the morbidity and mortality of children is tremendous. In this case, it's very, very small. Yes, there are deaths in children, but almost always those deaths are associated with comorbidities, other diseases in the children. Interestingly enough, one of the prominent comorbidities in otherwise healthy children that leads them to worse outcomes is the presence of obesity. This is true in adults, and it's actually true for many, many diseases in children who end up in a pediatric ICU. Another question that we have to address is, will children and or adolescents become infected and sick as they re-aggregate in schools? And this is, of course, is the great fear. What is the risk for school personnel? If the teachers become sick, if the school personnel become sick, then schools cannot stay open either. Can K through 12 students spark outbreaks not only in the school, but throughout the neighboring community? And finally, how do factors such as socioeconomics, race, ethnicity, and special needs of some of our students affect COVID-19 as schools reopen? So these are some of the key issues that we need to address and that a public university should deal with as we consider this question. And I just wanted to point out, these are some of the perhaps unexpected consequences that occurred when the world decided at the beginning of the pandemic to close schools. And the problems that we see in children are obviously interrupted learning, poor nutrition. We're starting to see increases in obesity in kids who are unable to be physically active because there's no school PE that they can have. The high economic costs, the unintended strain on the healthcare systems, the rise in dropout rates. There's an increased exposure to familial violence, unfortunately, and exploitation. And the social isolation many parents will tell you is really quite remarkable. So here are 4 schools that we're dealing with, and I think they present the kind of a picture that we are all facing throughout the country as we consider reopening the school. So here's public school in Santa Ana. It's in an area where there is a surge underway, which of course, as Dr. Monuki pointed out, is starting to abate, but may return. And unfortunately, I've already spoken to a number of principals in Santa Ana, and there's already a case of COVID-19 orphans where both parents contracted the disease and passed away. This is another school we're dealing with. It's a charter school in Santa Ana. 80% of the students in this school rely on the school for food security. And many, many school personnel in Santa Ana have literally spent the last 3 or 4 months driving around and delivering food to students in need in their communities. The tales that I think will emerge about the altruism and selflessness of teachers and school principals and others is really, really remarkable. Our third school is a charter school in Orange. This is a really unique school because it takes care of students with special needs. For example, autism spectrum disorder and Down syndrome. I want everybody to think about what we mean when we say physical distancing in the context of the school. So it's one thing with developmentally appropriate children who are able to understand the basic idea of physical distancing. But here, these are kids for whom if you suggest, for example, hand washing or even going to the bathroom, in many cases, they need to be accompanied at very close range by an aide or by a teacher. And so the kind of PPE and the kind of recommendations that we need to make for these schools to open is going to be very, very different than other kinds of schools. And then our fourth school is a private school in Newport Beach with a population of predominantly middle and upper middle class families. And if you look at Orange County, you may know that when Governor Newsom set the original plan for criteria that would permit schools to open, there was a mechanism that schools could get waivers. The schools in Orange County that got waivers, except for one or two exceptions, were private schools. This particular private school in the past 4 months has spent $1 million to prepare for reopening. And this is an amount of money that has been out of the range of even the charter schools and certainly the public schools. And we've had many conversations with these schools about the need to advocate and to change the situation so that they can meet the demands of reopening. So what can our UC team contribute? Well, in my own case, in my own research group, we've had many years of NIH funding that's been focused on health in schools. And my own research area is in physical activity and exercise. And we've been fighting in schools a different pandemic, and that is obesity and type 2 diabetes, which hasn't gone away. And about 12 years ago, we had the great good fortune to be one of the sites of the largest NIH-funded study ever undertaken to prevent obesity and type 2 diabetes in predominantly lower socioeconomic schools. And this was called Project Healthy. And we literally turned the schools into laboratories. We set up so that we could do oral glucose tolerance tests in the school gyms. And we processed hundreds of children. We followed kids between the 6th and the 8th grade. And this gave us a tremendous understanding of the challenges faced by schools and what one would have to do if we were to do a study in a school focused on the COVID pandemic. The results of this study were published a number of years ago in the New England Journal of Medicine. And so at least we had experience in how to deal with any kind of study that would be done in the school. The other thing we can do is sound the alarm. Several months ago, we were able to organize a group of really prominent and well-known pediatric scientists, teachers, epidemiologists, and we wrote this commentary that appeared in the Journal of Pediatrics, Reopening Schools Safely, the Case for Collaboration, Constructive Disruption of Pre-Coronavirus Expectations and Creative Solutions. This was meant to be a wake-up call. We knew that the challenge of reopening the schools would be tremendous. We didn't realize at that time how political this whole issue would become. One of the things we do as a university is we plan innovative and necessary research. And we did that, and we have funding to do a project which was just approved by the IRB and is funded by Orange County Healthcare Agency, by Children's Hospital of Orange County, and by UCI. We have two fundamental aims in this study, which we're going to do in the schools. We're going to measure SARS-CoV-2 transmission in students and personnel in selected schools as they reopen. And not only are we going to look at this, but we're going to try to focus on some of the basic virology to understand why is it that children seem to have fewer symptoms than adults, the epidemiology, and a very important goal is to look at the impact of socioeconomic status and race and ethnicity. In this diagram, you see the difference between a pediatric lung, this is an alveolus, the air pocket in a lung, and an adult lung. We need not go into details, but there are a number of theories out there to try to understand why it is that children have less symptoms. And this has really important implications for vaccine testing. So because the disease is so much more mild in children, unlike a disease, as I mentioned, like polio, we have to be very careful about vaccine testing and the ethics of vaccine testing in children, where we weigh the risks and the benefits of any kind of untested vaccine, which, as I noted, in children is much milder disease. Another aim is to measure the fidelity of mitigation protocols employed by the schools. Now, we all know what we're supposed to do. We're supposed to do physical distancing. We're supposed to wear masks. We're supposed to wash our hands. But how do we study this? And so we adapted a novel approach that had been used in my field in physical activity called the System for Observing Play and Leisure Activity in Youth. We got together a group of scholars from around the country, and we added to this direct observation a look at things like face mask wearing and physical distancing. I'm going to make just two brief comments about face mask wearing. The below the nose community has been stunned because the study shows that the nose is indeed connected to the lungs. Every one of us have seen this way to wear face masks. In Orange County, if you point this out to someone, you definitely incur a risk of bodily harm, so I wouldn't do that. And I also want to point out that young children can have fun with face masks. I've been to Montessori schools where 4- and 5-year-olds wear face masks, and they can be seen to have a good time with this. And so this is not an impossible task. And here's the flyer for our research study, which is sponsored, as I said, by CHOP, UC Irvine, and Orange County Healthcare Agency. And we're about to start this study in a couple of weeks in one of our schools. The university can also work in partnership with our schools in a number of areas. And this is just some selected challenge areas and what a university can do. For example, content expertise. School principals and school personnel are wonderful in the knowledge that they have about education, but they really are worried about the medical, biological, epidemiological aspects of this disease. And university can provide access to a variety of content experts. And Eileen mentioned the UCI CHOP Healthy School Restart Working Group. And we work with many schools to give them our best knowledge about a lot of the medical information that's coming. Information assessment. We can do rigorous literature review and continuous updates on the COVID-19 clinical issues. And we can help with startup plans and monitoring, review and provide feedback to individual schools as they develop plans for restart. Little has changed since this pandemic began about our understanding of what we need to do from the 30,000-foot level. What the schools need is help in how do you implement face masks in a school in Santa Ana? How do you do social distancing in a crowded school in Orange? These are the kinds of very specific issues that must be addressed, and it's logistics, logistics, logistics. And I can tell you the schools are so grateful to us. This is just a picture that appeared on the Instagram page of one of the schools in Santa Ana. And the principal told us this was 3 of our personnel, Dr. Ulloa, Diana Stevens, and some of the other personnel. Just the fact that we visited the school made the parents feel that the school reopening can be done safely, and that the doctores were there to check the school out. And we can assist school districts to evaluate the data. In the past several months, we've witnessed a tsunami of data, information, misinformation, truths and half-truths, all related to the school restart. Just for an example, and this was originally used by the administration to say, oh, there's no problem with reopening the schools. And they showed the data for Norway. This is the number of cases. This is when the shelter at home began in that country. This is when the schools reopened partially in that country. And they say, look, there was no effect on the overall cases. But what they didn't show you is this data. And this is from Israel. Again, you have the shelter in place. You have the number of cases, the schools reopened. And I'm not saying that this is causally related, but at least in this country, there was a huge spike in the number of cases in this society as a whole. And there is just a tremendous amount of confusion in the data. This was July 10th. Children rarely transmit COVID-19. Doctors wrote in a new commentary, schools can reopen in the fall. And then a week later, in the New York Times, older children spread the coronavirus just as much as adults. Large study finds school reopenings will trigger more outbreaks. So we can at least help the schools sift through this amount of data. So what is our bottom line? The take-home message is that schools must be perceived by parents as a healthy place and by teachers as a healthy place. And the UC system can play a key role to ensure the healthiest possible reopening of our K-12 students. school personnel and families. And to achieve this, we can simply follow these ABCs. We need to activate our researchers and scholars. That's one of the reasons that we're doing our study. We need to build bridges of trust, team science, and continuous learning with our school personnel and partners. And we need to collaborate across California's diverse communities. And I found this quote from Albert Einstein: as for the search for truth, I know from my own painful searching with its many blind alleys how hard it is to take a reliable step, be it ever so small, towards the understanding of that which is truly significant. I found this picture of Albert Einstein at a family reunion of mine several months ago. And it turned out that this was a picture taken in 1921. And this gentleman on the right was a school principal in Israel and my great uncle. And it reminded me that we have this obligation to the next generation, from the university's perspective, taking care of our K through 12 students. These are going to be our students in the years to come. So thanks a lot. I look forward to your questions.
Thank you so much, Dan. With that, again, just one last reminder, we have one more speaker and then we'll be open for questions across the board. I know there are a number coming in that people are answering on the side, but I think we have some that are going to be critical that we'll get to. With that, I'd like to introduce Dr. Daniela Bota. Dr. Bota received her MD in Romania, but her PhD from the University of Southern California. She was a fellow in neurological oncology at the Duke University Brain Tumor Center. She's a professor of neurology here, associate dean for clinical research, vice chair for academic affairs in the Department of Neurology. Most importantly for my life, she is the clinical director within the Sue and Bill Gross Stem Cell Research Center, where she's done a phenomenal job in terms of working with our pipeline for clinical trials related to regenerative medicine. And in this particular global crisis, working with CIRM and other organizations from the perspective of how we can bring our new tools and our research to bear towards clinical trials. So Daniela, welcome, and thank you very much for making the time. Please go ahead.
Thank you for your kind introduction. And hopefully we can have the system working and working appropriately. So maybe many of you are wondering why we're having a discussion about stem cells in the context of an infectious disease. And the reason for that is that CIRM does not support only primary stem cells. A lot of the work that has been done is to support any type of cellular and biological therapies. And when we think about the viral mechanism that you have heard from the speakers before me, what you know is that the cells, the viral cells, are always trying to infect the normal body cells. So what the antibodies can do, they can block the viral fusion. They can stop the virus from entering the cells. Now, the antibodies can be generic antibodies, like many levels of antibodies that we make in response to the virus. This is where the donors, the plasma donors, will come into the discussion. But we can also make very specific antibodies that will block specific molecules on the virus. Or we can make antibodies that can block the inflammatory responses that end up with the pulmonary inflammation and with the people going on ventilators. We also have immune cells that can be collected from the blood. They are called dendritic cells, and we can use them to form dendritic cell vaccines, which hopefully will address the question of how not only generate immunity to the virus, but how do we maintain the immunity over longer periods of time. COVID-19, it's about destruction as well. So you can imagine then why a number of the projects previously sponsored by CIRM for other indications are now focusing on COVID, because we can use the power of stem cells to repair the pulmonary damage and other tissues and induce tissue repair in order to shorten the convalescence and being able to bring people to a better health. I'll start with the convalescent plasma projects. Many of them at our institution are funded by CERN. And the goal of this project is to collect plasma from different donors as much as we can, and especially to collect from people with different backgrounds from different communities. There is a lot of discussion about how we can use the convalescent plasma, and many of you have seen the press release saying that now FDA allows convalescent plasma to be administered as standard of care. Experimentally, which is the line of work on which we can use the plasma three ways: for clinical trials, for single patient help, that's single patient INDs, and for the expanded access program. The reason to do this is to learn more and be able to answer the questions that are still unanswered about what is the optimal level of antibody a donor should have in the blood in order to be an efficient donor and protect future people from infection. And also to know if we treat a certain patient that's infected, what is the best stage in the disease at which the antibodies donated by other people can be used. So we have discussed about how antibodies are generated. We generate antibodies to almost each infection. If we have the ability to transfer the immunity, this is passive immunity transfer. It could be a way to bridge until the time that the vaccine is universally available, or to be able to treat people that for different reasons, very limited reasons, cannot be vaccinated. So listening here today, I'm also going to ask you to consider becoming part of this research. We need people that want to donate a small amount of blood so we can learn if they had the infection. And if they did, we want them to donate more blood and plasma in order to develop a convalescent plasma bank at University of California, Irvine, and part of the CERN efforts in the whole California in general. The other type of cells that we talk about are the NK cells. NK cells are immune cells, and we are the first center in California to open a very interesting study where we use cellular therapies, natural killing cells, cells that can kill the virus and protect from the infection, and they are derived from human placental cells. The preliminary data on this study suggests that we are able to transfer those cells to the patients that are sick with COVID-19 and admitted in the hospital, and potentially prevent them from being worse and going on the ventilator. What about repair? This is where we discuss about mesenchymal stem cells. And this is a project from Pluristem, a company that we partnered with in the past to do neural stem cell repair for stroke cases. Now we are using a very similar technology to try to modulate the immune response with the mesenchymal stem cells, as well as to try to see if we can induce pulmonary repair in patients that already have pulmonary damage and they are already on mechanical ventilation because of the COVID infection. Another interesting way to use biological materials to fight COVID-19 is to define a diagnostic system that is portable. Our collaboration was a company called Diomics. It's starting the work to generate patches that travelers can receive and wear before going, by example, to an airport. The patch will change color if there are proteins from COVID-19 in the blood. And when people will cross through a screening test, here it's the airport, the person that is positive that the patch changed the color will be identified, And we'll have the ability to do a very fast test to confirm or infirm the infection. While the people that were negative and for symptoms, as well as for the changing color of the patch, will be able to continue their flight, knowing that they are having very small risk of infection. This is extremely exciting for us. We were going to pioneer both the preclinical development in the lab of Dr. Lakey and the clinical testing and clinical trials in our alpha stem cell clinic. So hopefully, if we have a vaccine and we have a way of detecting what the people that are infected, we will be able to open more of our schools and visit our parents in the nursing homes and go back to a more normal life. We are doing many other things at UCI on the stem cell world, but the last project that I want to talk about is the vaccine that we have in the plans to open at UCI, together with one of our local company, Ivita Biotherapeutics. And why I'm talking about this vaccine is because right now the vaccine development, it's very exciting, and we have a number, in the US, we have a number of clinical trials coming to the phase 3. There are also questions about the product in England, and maybe some of you are aware also about the fact that Russia already approved the vaccine, though there are not all the stages of testing completed. The problem with many of those vaccines is that they don't confer prolonged immunity. And as you heard from Dr. Tan, people can reinfect. So one way to overcome this is to create a reservoir of cells in your body that will continue to stimulate and produce immune cells, and those cells will be already educated to fight the infection. So this is the process that it's proposed here, where a small amount of blood will be separated, the immune cells then will be exposed to viral particles in the dish, purified back, and then given as a set of injections to first-line personnel, people at high risk, in order to protect them from having additional events. We will open this study very soon. We're expecting in the next 1 to 2 months, and we are hoping to enroll 180 participants. So again, if you're interested, please let us know. We think that this is going to be able to move the science. I don't want to take more time. We are very grateful that you stayed so late with us, and I'm happy to take questions.
Thank you so much, Daniela. I would like, just before everyone in fact leaves and we get to the next set of questions, to just show one quick slide. So I am going to share my screen for a moment here so that we have the opportunity, in fact, to thank not just all of our speakers, but in reality, the other key participants in terms of making this work tonight. And that is really a number of people in addition to the speakers. First, Sue and Bill Gross, who without their generous support, we wouldn't have a stem cell research center from which we can host these type of events, including our community lecture series. I have a feeling that our Zoom webinar chat is showing up. Brian Cummings, who organizes the community lecture series, is one of our valued faculty here at the center. Judy Beck, who handles our outreach efforts and coordinating. Will Alvarez, who was a senior producer with UCI Media, and Kyle Good, who also is a videographer and media producer here at UCI, who has been incredibly important in terms of our community lecture series. So thank you very much. With that, if all of the speakers could join me, so to speak, by turning on your video, what we'd like to do is be able to field several questions. And I know a number of questions have been answered on this side. I'm going to do the best I can to moderate here that have been coming in. So, but not everyone, just so that the speakers are aware and the audience is aware, not everyone can see those. So if someone is logged in through Facebook Live, which we have a fair number of people, they're unable to see those questions or answers. So there may be some repetition. Please bear with me in that regard. I'd like to start out with this question. And Ed, I'm going to hand it off to you. It compiles a few things together from what we've seen so far. And really, if I lump it, it's where is the right balance between speed and sensitivity? So with quicker, less expensive tests coming down the pike, do we have a balance that we can reach where an expensive, very sensitive test maybe could be too slow to really come into play to do a good job in terms of slowing down spread? Where do we hit the right balance in terms of what types of tests are employed and the new tests that are coming that may be faster, but potentially not as accurate or not as sensitive?
Right. So I think you really hit the nail on the head. It's all about trade-offs. You're trading one good feature for another good feature, and it depends on the particular context and the particular problem that you're trying to solve. And I think right now we're trying to solve it. I think it's less about having better and better quality tests, but now having more tests that are available that can turn around a result quickly, that you might be able to, afford a loss of sensitivity. For example, with this mass asymptomatic screening at UCI, the idea would be you need a lot of throughput. So the number of tests you can do in a certain amount of time. You need to be able to do a lot of those. So the throughput of the test is important. The turnaround time of the test is important because the faster you can get the result to the contact tracers, especially the positive results, so they can investigate whether it's truly an infectious case or maybe it's somebody who is actually convalescing from an infection from 3 months ago and they're not contagious and they don't have to worry about it. The faster we can get the result, the better for them. And so I think that the country, especially with the shortages and the need and the desire to get more and more testing, is getting more and more comfortable with the idea of having less accurate tests, but maybe ones that could be implemented in a way that could meet some of these other goals that we would have. So you're absolutely right. It's all a trade-off, and evaluating the trade-offs for where we are and what we would like to do and what we need to do.
Right, perfect, thank you. With that, Ming, I'm gonna put you on the spot and do kind of the same thing, compile several questions sort of together. A number of discussions that have happened on the side and questions that have come in have been focused around the idea of our ability to generate vaccines is certainly probably better than it was in the 60s, but maybe we don't have a vaccine for the common cold, for example. And there is some data that there's a certain mutation rate, as you noted as well, for SARS-CoV-2 that could get in the way in terms of vaccine development. So how much of an issue is that? And will it in fact be possible? Do we really have a good feeling at this point that it's going to be possible to develop one or more vaccines that can be deployed in order to be able to start to really open things up without the same kind of concerns that we're all struggling with right now?
Ming, you're muted.
Yes. So I was
Yeah,
saying that it's a very important question.
And here I thought it was just me. Can't hear anything.
It's an important question because a lot of discussion is about when are we going to get a vaccine as opposed to can we get a good vaccine. And vaccines have been tremendously helpful in decreasing and sometimes eliminating infectious diseases, but we don't have vaccines against everything. So as you mentioned with the common cold, we don't have a vaccine against that. The good news is that there is reason to believe we can get a COVID-19 vaccine. There are related viruses, SARS, and also the MERS virus, where some vaccine work in primates, so in monkeys, has shown that you can elicit some protection. We don't know how long the protection may last. It may be in the order of several years. But it seems as if it might be possible to have a COVID-19 vaccine. I think one of the issues we're going to get is even if they announce a vaccine, the question is how good is that vaccine? And you'll have multiple competing vaccines, so we may face the choice, which vaccine do we get? I think that you asked a question about mutation rate. I think that's something we'll get to later. First of all, can we get a good vaccine? And secondly, like influenza, will it change over time so we need a new vaccine to keep up with it? That's an issue for later on. What I would say is all the rosy projections of having a vaccine by the end of the year, maybe you'll have a data showing the vaccine works. They won't have large-scale production of a vaccine by then. I want to say one last thing about the scale of the project. Most vaccines, all vaccines that we have in the country, except for the flu vaccine, involve 5 to 10 million doses a year. That's usually to vaccinate a cohort of kids a certain age. The flu vaccine is the exception, okay? And it's a tremendous effort to have a dose for everybody. COVID-19, we're talking about making vaccine for everybody on the planet. We don't have the scale-up facilities to do this. So even if you had a great vaccine, making the vaccine, having it be, the distribution and all that, that's going to be a major issue. So when people say we'll have a vaccine by the end of the year, I'm doubtful. But I hope that by then we'll know whether we're on the right track.
Understood. Thank you again for your insightful comments. I'm going to follow up with one, though, as long as we're in your bailiwick here. We've had a number of questions about herd immunity and just a general understanding of what is herd immunity, and is it something that is actually achievable? Are there examples of other types of infectious disease where we have achieved herd immunity over time without a vaccine, and how long does that take? What does it look like?
Right. So herd immunity is this concept that if you have enough immunity in the herd, in the group, then you don't have efficient spread of the infectious agent. And that means that even susceptible individuals may not get infected because there's not enough of the agent to spread around. And you can develop that immunity through natural infection, or you can also develop it through vaccination. And we have seen that for a number of childhood infections, once we had a good vaccine, the rates just plummeted, and even kids who didn't get vaccinated didn't get the natural infection. And so some people have talked about this freeloader effect where some kids are not vaccinated, maybe their parents don't want them vaccinated, but they derive protection because all their peers are being vaccinated. And so one of the questions is, what level of immunity do you need in a group or population to have that collective herd immunity? And typically they talk about 80 to 90%, but there've also been some studies and reports saying that in certain scenarios, you don't need that high herd immunity. People have even floated ideas of 40 to 50%. And I think that the way to look at this is you may not need to vaccinate everybody in the population. There may be certain people involved in the spread, and if you can vaccinate those individuals, then you can interrupt the spread. There's even been speculation that in places that were hard hit in the early days, like Northern Italy, near Milan, or New York City, maybe they've reached herd immunity, because what we can't explain is why did they have an outbreak and why did the outbreak go away on its own? So maybe that was an example of local herd immunity. But the thing is, we haven't done the widespread antibody testing to know what the level of immunity is.
Can I ask?
Okay, thanks.
Arlene?
No, go ahead, because I was going to riff to you, Dan, so go ahead.
Let me ask you a question, Ming, because this comes up a lot, and I think you should comment on this, is some people think, well, why don't we just let the virus go? Why don't we just let it happen? We'll have herd immunity and everything will be fine. And I think I know the answer, but I'd love you to address that, because sometimes people bring that up.
I think there are a number of issues. So one is that you could let the virus go, and we know already people will die, and vulnerable people will die. And so one of the things is, are we prepared as a society to take so many losses? I mean, we have almost 200,000 people who have died in this country. This is no small number. From a normal influenza season every year, the number of cases directly from influenza and secondary effects is about 50,000. I think there are also about 50,000 motor vehicle fatalities a year. 200,000 is a huge number. I think another thing is we don't know yet about immunity. So you could say, well, let's let everybody be infected naturally, but we don't know, maybe you're going to get second and third infections. And I think another issue is we don't know enough about complications. And so, there was initial thought about, well, let the children be infected, but we're also seeing that a number of children have complications of death. So my own personal view of this is these are relatively early days. We don't know enough. I'd like to know a lot more before I put myself at risk. So if I can delay getting COVID-19, I might improve my odds because I'll know more about what the risks are. Famously, Sweden tried this approach, and compared to its Nordic neighbors, it turns out that they had higher cases, more deaths, and the same economic damage. So their neighbors around them don't think the Swedish approach worked.
Right. So let's turn the tables a little bit, just in the interest of time. Dan, I'd like to bounce over to you for a moment. We have a number of questions that cover the general kind of theme of with kids going to school and schools reopening. You showed the data on this from Israel, for example, in part. We can expect that potentially asymptomatic kids will be coming home, bringing that virus back to their families. How should families be thinking about managing that? Do you have advice for them? And then a parallel set of questions about risk factors, really, and people that are concerned because they fall into a higher risk category for a variety of reasons, diabetes, for example. And when will it be okay for those folks to be thinking about going back to work or moving out of what's a completely remote work environment where everyone's been living for the last 6-7 months or so?
Thanks. So, you know, the first question is a complicated one. The overall approach I think that we have to take is that we are not, as Ming pointed out, and as everybody's pointed out, we're not making this virus go away. I'm glad, Ming, that you pointed out that, you know, even if we have a quote successful vaccine, by December, all we'll know is that people may have had antibodies for 3 months. That's all we'll know. We really won't know the long story. So we've got to decide how we're going to live with the virus. And this is really important when you consider opening the schools. So assumption number one is if you do open the schools and kids come back together, that they'll spread the virus among each other and ultimately to teachers. And as you pointed out, Eileen, maybe they'll go home and spread it to adults who have comorbidities at home. And by the way, this is one of the huge areas where the socioeconomic, racial, and ethnic disparities in this disease have really come to light. In Santa Ana, which is not very far from where we are right now, there are many, many people who live in multi-generational families. There's a grandma with diabetes. There's a grandfather with heart disease. There are, you know, 700 square foot home with 7, 8, 9, 10 people. And the ability to spread from a child there is obviously much greater than a home in Newport Beach, which is 3,000 square foot and 2 relatively young parents. So assumption one is you bring the kids back together, they're going to spread the disease. Assumption two is the one that we don't often think about, which is that won't happen if we don't send the kids back to school. Ming, you pointed out that the increase in pediatric COVID has been greater in the last 3 or 4 months since it was at the beginning of the pandemic. And schools were out during that period of time. And so I think of in Orange County and in LA Unified, 1.2 million K through 12 children and teens roaming around. And if you think that they're not going to interact with each other, that's not the case. They are going to interact with each other. Now, what about assumption one? Well, the European experience and the Asian experience suggest to us the following: that if the school is able to put into place really strong mitigation procedures, face masks, social distancing, cohorting the children, you know, hand washing, you really can minimize the spread. You can't eliminate it, but you can minimize the spread. So I think we can do that. And I think that what the society has failed to do over the past 4 or 5 months is really help particularly the public schools and particularly the lower socioeconomic schools prepare for this. And that to me is almost heartbreaking that we haven't been able to do that. As far as, Eileen, your second question, you know, so the context in which I think we're all aware of this is somebody like a school teacher or an essential worker who has a comorbidity. Do you expose yourself in any way to the risk of getting this disease? In the case of schools, for example, I would think that a teacher with a known comorbidity, as well as your school might try to prepare, we know children, they're going to come up, they're going to hug you, they're going to take their face masks off. And so I think the attitude of the schools, at least, has been that we're not going to force any teacher to come.
Right. So can I just do a quick follow-up with you? Because I think actually you are the right person to address this question here. And that's a parallel that's come up online actually as we've been chatting. Because of the, let's say, the wide array of information that we all find, messaging is very different, federal to states, local health organizations, state to state. So I think one issue that we all see is people, ourselves included, are really grappling with trying to filter that information and get to the right stuff. So Dan, where is, at this point, do you think the right place for folks to be able to get their information? And a follow-up to that would be, how do we counteract, or should we be thinking more about counteracting the spread of misinformation that's out there? Because that certainly has been probably an inhibition to being able to do the best response that we could have out of this situation.
Yeah, so those are great questions, Eileen. And I would only add one other factor, which is the politicization of almost every aspect of this disease. And I don't know how to address that. That's above my pay grade for sure. I showed you the diagram that had been used by the administration to say, you know, their tune was very different about 4 weeks ago. They were saying, just open the schools, send kids back, there's no problem. They realized that that was not a good argument, even if you're in favor of sending kids back to schools, as an example, because people are not that stupid and they know that you've got to have some kind of mitigation. I think that the only answer is that, at least for us as universities, is number one, Eileen, what you guys are doing right now, this kind of a meeting where you give the public the opportunity to interact with, you know, people like Daniela and Ming and Ed and you. We don't have all the answers, but we at least can point out some of the egregious errors that we see, and everybody in this group has done that. In the case of the schools, we have interacted with the Orange County Department of Education. We're on the task forces. And I think in every industry, our university has done this. And we should serve as a reservoir for helping folks to look at the information in an intelligent way as possible. And again, this kind of meeting that the Stem Cell Center has done is a really wonderful example of how we can address the issue that you raised.
Well, thanks. And thank you again so much for participating. Daniela, if I could turn over to your court for a moment. I think one question that clearly is relevant is what do you think, in so much as you can comment, of the current clinical trials that are ongoing? Maybe just comment about how quickly this has moved, really, and where do you see the potential of those trials being, you know, in 6 months or in 12 months as this rolls out, as we've heard from Ming and Dan and Ed, that we are in this for the long haul right now.
Thank you for addressing this question, Eileen. And when we look at clinical trials and clinical development in general for COVID-19, we are able to see a speed of development that we have not seen before. This is truly unique. I've been doing clinical trials for 20 years. I don't think that I ever have seen a response that is so strong, that involves so many clinicians, so many scientists, and such a unique focus. And this is not only at UCI, but it's across United States and in reality across the globe. I do believe that we're gonna get some answers, and we already are having some answers. We had the Remdesivir approved. We are learning more about the convalescent plasma. Very soon, as Dr. Tan mentioned, there is going to be at least one vaccine that's going to look like it can prevent maybe the high rate of contamination. But the disease is here to stay. So now we are changing from having 2,000 companies trying to develop diagnostic devices and many of them failing, to having a few of them that are able to move forward and produce really solid results. So I think that we're gonna see wonderful things in 6 months, 12 months, 24 months, and it's hard to predict when we're gonna complete research. Dr. Cooper, do you want to comment from your experience? Have you seen a response similar with the response to COVID-19 before?
No, and we really need to give credit to Daniela, to your organization, CCR, and really boosting the numbers. It's really remarkable. I think you relayed to us last week that there are 14 or 15 ongoing clinical trials just at UCI. One number I think, Daniela, that everybody would be interested in, and we had the statewide call across the UC system. So even though we had seen an increase in the numbers of hospitalizations, our death rate continues to go down. Now, there's who knows why that is. It could be because we're better at treating it. We know how to use the ventilators better. But I asked Carrie Byington and I asked Atul Butte, who are our leaders at UCOP, wouldn't it be interesting if we saw that there was as we saw the death rate go down, I have a feeling we're seeing an increase in the number of these COVID patients who are enrolled in these clinical trials. And there's going to be some efficacy in these clinical trials. And so I have this feeling that we're already seeing a result of novel thinking, innovative thinking. And at UCI, and Daniela can certainly address this, we go from remdesivir, which is an antiviral, to novel anti-inflammatories, all across the spectrum of this disease. Maybe, Daniela, you could comment on that.
Thank you for introducing this concept. And indeed, we are talking right now about hypotheses and sporadic results. But one of the things that we are very proud when we talk about UCI results, and you see heroes work here, and we are not the hero, but the frontline workers are the heroes. However, scientists come very close behind because we are trying to make the work better and better. And why I'm saying this is because we know that mortality from COVID-19 at UCI, it is very low as compared with other California hospitals, up to 50% lower. And this might be because of our exceptional physicians and our wonderful equipment, but it's also because a high percentage of our patients have enrolled in clinical trials. I think we are right now around 120 patients that participated on the interventional studies. And many of them, through those studies, had access to drugs that otherwise would not have been available. So the more that we do, the more that we have large databases accumulating results across the nations, across California, and hopefully across multiple countries, we will learn more through doing this kind of population analysis. What are the true breakthroughs that we are seeing?
Fantastic. And can I just press you also for one follow-up, and then I understand it's getting quite late. It's 8:42. So I have kind of a unifying question, I hope, for everyone just at the end, and we can think about shutting down for the evening. So one question, actually maybe two, I apologize, that have come in that I think would be important for people for general information is about the safety and what's known for convalescent plasma as a therapy. Sorry, I think that goes to you, Daniela. And then really kind of a parallel question for whoever thinks they would be most appropriate to pick that up. I know when I look on Amazon, you see a lot of masks that have copper or different sorts of alloys that are infused, all kinds of advertising out there. Is there any efficacy data that would support that would be useful for people to have information about? So please.
So I'll take first the convalescent plasma and the safety question. Of course, I'm gonna need at some point to involve Ed Monuki, so Ed, please be ready. Convalescent plasma collection is done in the same conditions on which we collect blood, with extensive testing for any kind of viral infection, any kind of bacterial infection, with extensive HLA typing. So we make sure that the plasma that is given to patients that need it, it's truly matched to their blood type. We have an excellent transfusion center, which lives in the Department of Pathology, and they have done this kind of cellular processing for years. So this is a very, very safe process. Now, how effective it is, it has been effective on other diseases, and the recent data presented suggests that it's effective as well in COVID-19. Dr. Monuki, do you want to comment on the transfusion medicine and plasma collection?
Right, thank you, Dr. Bota. I totally agree. Convalescent plasma has been used for so many different indications in the past. So we actually have a good understanding what the side effects are or the adverse events, and it's quite low, generally less than 1%. You can have some severe reactions, allergic, transfusion-related injuries, but in general, the complication rate is quite low. So it's known to be a safe therapy in general.
Do you want to comment on the face masks?
Yeah, so I was just going to redirect face masks.
Yeah, so here's what we know. I don't think there's any data about copper or materials like that. I think what we know generally, if you look at the good websites like the Johns Hopkins website and some CDC, is sturdy cloth face masks of multiple layers, like good cotton ones, will do the job of preventing the droplets, which is really the most important thing. The ability to prevent the virus itself is probably the actual tiny virus. The microscopic, submicroscopic virus is probably not the problem because the virus is carried on these aerosol droplets. Surgical face masks, if they're available, and they're increasingly available, are useful. None of this stuff is completely fail-safe. We know that, but we do know from studies that we've done at UCI and across the UCI system that when our hospital workers, our frontline workers, wear PPE, and they're dealing with patients who are known positive, that the rate of infectivity, and Ed, you can address this, is very, very, very low. The outbreaks that we've seen here is when our personnel forget that they can't just take a break, take off their PPE and have coffee with each other. But when a face mask is worn correctly and it's got a decent material and you do it in public correctly, it provides a great deal of protection. Ed?
Yeah, completely concur. I think the good news is that a lot of the masks that are now more readily available are quite effective, especially if both people are wearing the mask. And as you said, the number of sort of secondary infections occurring in the healthcare setting has really been quite low. Where they have seen it is in break rooms, where again, maybe you relax, and of course you're eating something, so you can't eat through a mask, not yet anyway. So, which is why I know our infection prevention team is focusing a lot on the break rooms there. So, but in general, the masks, fortunately, are quite effective. Now, somebody in the Q&A mentioned bandanas and gaiters and those things. And that's absolutely true. I think people who've studied that, those are not as effective as a sort of quote unquote real mask.
So I'll just maybe, although outside my area of expertise, but one resource that people might find useful is a nonprofit called Masks for All, MASKS for the number 4 ALL, which has really, I think, some very easy to understand transmission data. And
missed a little bit of your answer.
Sorry, as I was telling my colleagues on the side, I am on the UCI campus in my office, so my internet would be good, and it could be better this evening. So Masks for All, MASKS for the number 4 ALL, is a website that you can go to. It has some really easy to understand information about transmission and why masking is so important in terms of controlling the pandemic and where we are right now. So if I could, assuming that came through all right, I'd like to ask everyone a summary question and ask each of you to reply, and it's a pretty simple one, really, but it, I think, captures a lot of the uncertainty that all of us, and I think the people who are tuning in tonight or will be over the next few weeks, are feeling. On one hand, we can say that this could have been a much worse situation. We could have had a much more lethal virus, novel virus that was released on the world this year. But we certainly had a lot to learn. And so a good thing potentially to come out of this are all the things that we are learning going forward. So my question is pretty straightforward to each of you. Daniela, to you first. What have you learned from this? What have we learned from this collectively going forward that is going to help us? And where are we going to be in a year?
I think we have learned how to work together. Institutions and organizations that have not worked together before learn by necessity that they have to change their ways and pull in the same direction. I think we also learn that there is not such a thing as too much education. And maybe we have not done enough as a scientific community to educate. And should we in the future have a similar event, I think a lot of us will be much more verbal about making sure that the correct information is spread and that the community is informed about the appropriate way to protect their health.
Thanks. Ming. Ming, you need to unmute.
How the virus is spread. So in fact, I think we have the tools to interrupt this. I think the thing to remember is that this virus is spread person to person. There's no other reservoir in the environment or animals. We could interrupt this if we all acted together. That's the trick. Not just protecting yourself or your family, but we have to do this as a community. But I look back on the lockdown we had, we wasted it. We all stayed at home for months. We could interrupt this with a 6 week strict lockdown, but we did a partial. Where are we going to be? I think that's the sad thing, because I don't see this ending by the end of this year. This is going to go on for a little bit. Let's hope we have better therapy, hope we have vaccines. But I think public health measures are going to make a big difference. We have the tools. We just have to have the will, and it has to be done at a community, at a national, maybe at a global level. Because I think what we're seeing is the countries, again, that had good success are having outbreaks again. This is not like a wildfire that comes in once. The tinder is still there. You put the fire out once, it's going to come back again. So we have to be vigilant. We have to keep working together.
Great. Ed.
Well, Dan may kill me because I'm going to politicize this also. But it's hard not to, for me to think constantly about the value of a central coordinated response and the downstream consequences that we all face because of the lack of a central coordinated response. They have just been so manifold and just in so many different directions. Just the competition between labs to get the reagents they need, the instruments they need, the pipette tips, and the fact that then the government will divert those en route to you because they've deemed a different site more important or a higher priority. Just that whole lack of coordination has made me appreciate the value of a central coordinated response in the face of an emergency.
Yes. Dan.
So yeah, I agree with what everybody said, and but I want to add one more thing. I think that the pandemic has really highlighted the inequalities in how people have access to health. We just see all of the numbers so different in communities like ours here in Irvine and literally a stone's throw away in Santa Ana and across the country. And I think one of the wake-up calls that we have to address as a society is it is not conscionable for us to have these levels of inequality in how one particular disease affects different populations. I think we've got to address that absolutely head on.
I want to echo what Dan said, because I think we've learned that our healthcare system here in the US works for some people, but doesn't work for everybody and doesn't work for the country as a whole. So I think that our fragmented healthcare system is one of the reasons why we have so many cases we haven't responded as well as the rest of the world. So after this is all said and done, I hope we have a discussion about how we can have healthcare for all. I think it's a basic right. I think what the virus has done is it probes and it finds the weak spots in your society. And as Dan said, the disparities, but our healthcare system that doesn't work for everybody is our weak spot. So hopefully we can fix that after, during, but after the pandemic.
Yeah. So we can, I think there's a conversation to be had to reflect on the cost of those inequities and that these things are very, very real. So with that, just so that you guys know from what I can see, which is not all of our audience, we still have over 80 people online. So I really appreciate all of your time. Thank you so much to the people that have joined us. Please share the video. It will be up on our website with people who need information. Reach out with your questions. We'll be sure to get back to you. Thank you for attending tonight. And once again, thank you so much to Ming, Ed, Dan, and Daniela for your fantastic and informative presentations, and to all of our organizers behind the scenes. So good night, and everyone be safe.
Thank you.
Thanks everyone.